Our next speaker is Amir Shlomai, who's going to address the new drug targets for HBV and some clinical data, and he's on the faculty of Sackler University in Tel Aviv.
Thank you very much. I would like to thank the organizer for inviting me to give this talk. And indeed, recently, there seems to be an increased interest in finding more definitive cure, more definitive therapies for HBV infection, with or without connection to the fact that in the HCV field, we are not standing in a curable disease right now, so why not to cure HBV as well? So where are we standing now, right now regarding anti-HBV therapies? So the standard of care for chronically infected patients who have, of course, indication for treatment is nucleotide or nucleoside analogs. And today we are blessed with the second-generation nukes that are very potent. They have an excellent resistance profile and also very minimal side effects. So if everything is so good, what is bad? The bad thing is that those nukes actually inhibit the viral polymerase, reverse transcriptase, but they do not touch the cccDNA that resides in the infected cell's nuclei. And therefore, there is no real clearance of the virus, and there is no cure. The other treatment option that we have now is pegylated interferon. And although the pegylated interferon works in several ways, through the immune response and through a direct antiviral, and the interferon alpha was also reported to have some effect, some more effect on cccDNA as compared to nukes, it's still quite far from clearing the virus and clearing the cccDNA in the cost of substantial adverse events. The combination of interferon and nukes seems to be rational. It has synergistic effect on antiviral effect, and several studies have tested the utility of this combination, either given concomitantly or sequentially, but they have not come to firm conclusions, partly because of methodological and other problems. So for now, in 2015, we cannot really recommend the combination of pegylated interferon and nukes as an antiviral therapy. So all in all, what we call or what we designate the closest definition to cure, which is surface antigen loss with or without the development of surface antigen antibodies, is right now infrequent in most patients. Lifelong antiviral therapy is often needed, and therefore it seems the better strategies to control or eliminate the virus are needed. So strategies to control the virus are broadly divided into those that target a step or multiple steps in the HBV life cycles, and those that modulate the immune response, which is a very crucial arm in an antiviral response natural history. Let me start with a few drugs or a few targets in the HBV life cycle. So, of course, the very first step in viral infection is the entry, and Stefan is already, and this most probably the best person to talk about it, has talked about the entry, this entry step. And of course, the NTCP, the discovery of the NTCP, the bile acid pump as a receptor for HBV, has greatly advanced our understanding of this crucial step in HBV entry. The virus uses the pre-S1 region in the surface protein in order to attach to NTCP, giving the rationale of developing the Mir-Cludex-B, which is a lipopeptide that mimics the pre-S1 and competes for HBV attachment to the NTCP. But there are other drugs as well, such as cyclosporin A and even bile acids that were shown to bind to the NTCP and compete for HBV entry. I will not go over the phase 2A clinical trial that Stefan has already talked about, but I would only note that although the HBV DNA was significantly reduced following Mir-Cludex-B treatment, there was no significant effect on surface antigen, at least at 24 weeks after treatment. So I would guess that in the future, those entry inhibitors will have to be used in combination, as Stefan also noted, with other drugs such as nukes, and maybe even more realistically, with immune modulators such as other drugs that would take care of the cccDNA itself and at the same time will prevent viral spreading. The other obvious drug target is the holy grail of the CCC, of the HBV infection, which is the CCC DNA itself. And for the sake of this lecture, I divided those strategies into four. One is pharmacological or small molecules, immune control, host factors that are responsible for CCC DNA formation, and gene therapy, and I will give one example for each. So, starting with the small molecules, this is a work coming from Haito Gao lab, and Haito actually used the HBV-19 system in order, and the unique feature about this system is that it expresses, those cells expresses the HBV both from the integrated HBV DNA and also from CCC DNA. But because of a special mutation that is inserted in the genome, the E antigen is secreted only from the CCC DNA. So you can use the E antigen as a surrogate marker for CCC DNA abundance. And using this platform, Haito screened more than 80,000 small molecules, and he found two molecules shown here that reduces nicely the CCC DNA level, most probably by inhibition the formation of CCC DNA from deprotonated RC, relaxed circular DNA. Another study coming from Ulla Protzer lab and published in Science last year have shown that by stimulating the interferon alpha receptor in HBV-infected cells, it results in activation of ApoBec3A, which is a deaminase, resulting in extensive deamination and mutation in the CCC DNA, leading to its degradation. But what was more striking in this a little bit controversial study was that upon activation of another receptor, which is called lymphotoxin beta receptor by agonist, you have got it results in activation of another ApoBec, which is called ApoBec3B, again resulting in CCC DNA deamination even more robustly than the consequence of interferon, resulting in a non-cytopathic CCC DNA degradation. And of course, things have to be further tested in animals and even in humans, but this opens the door to using those agonists as CCC DNA inhibitors in a non-cytopathic pathway, which is the ideal thing. Another study coming from Michel Nazal lab, published in PNAS, showed, I think for the first time, a host factor, in this case the DNA repair enzyme TBT2, to be a crucial element in formation and disconnecting the polymerase from the relaxed circular DNA, a crucial step in formation of CCC DNA. And what the authors showed was that if you knock down TBT2, you end up with reduction in CCC DNA. So here again, a drug target, a host drug target for anti-HBV therapy. Last but not least, the CRISPR-Cas9 system is very widely used today to induce mutation or to mutate the DNA directly or even to induce or to insert transgenes. And we, as well as others, thought that maybe it's a good way to design small guide RNAs against different regions in the HBV. And when we did it, and we injected it to mice that were hydrodynamically injected with HBV, we saw a very nice reduction in both surface antigen and HBV DNA. And even more importantly, when we induced HBV expressing cells with those small guide RNAs with the Cas9, the nuclease, we saw a very nice reduction in the CCC DNA, as is shown here in this southern blot after enriching for CCC DNA with HERT extraction. But of course, like everything in gene therapy, delivery is the issue here, and better delivery methods have to be developed before applying this method for clinical use. Talking about gene therapy, of course, we cannot skip the RNA interference introduced to the world somewhere at the beginning of this millennium. And many studies have shown that RNA interference against HBV is efficient both in vitro and in vivo. And it seems the HBV is maybe the perfect target for RNAi because of the overlapping nature of the HBV gene product. You can principally design a single siRNA which is complementary to this region, which is common to all HBV transcript, and knock down or eliminate all the mRNAs at once with only one shot. Another thing that has to be taken into mind is that the excess amount or the excess secretion of surface antigen is thought to act as a decoy for the immune response. And therefore, knocking down surface antigen might be a way to reactivate or to evoke a renewed immune response against the virus. And this is exactly what this compound, ARC-520, should be doing. And this is a phase 2 dose-ranging study for ARC-520 that was done on HBV patients who were treated with entecavir. And what the authors showed was that one IV dose of 2 milligrams per kilogram resulted in up to 50% reduction in surface antigen level, a reduction that was sustained for more than 40 days after injection. The other obvious target is the capsid of the core. And given the many actions that the core and the capsid have in the HBV life cycle, of course, packaging, cccDNA maintenance, and even expression from the cccDNA, and even ISG, interferon-stimulated genes response, so one can expect that targeting the core or the capsid will result in robust antiviral effect. So there are many compounds that are supposed to deal with the core of the capsid, and generally they have a few mechanisms of action by decreasing the core half-life, inhibition of pregenomic RNA packaging, and preventing the capsid formation. Here is an example of such a compound, NVR3778, that induces the formation of abnormal capsid, and this compound has been shown both in vitro and more recently in vivo in UPH skid mouse to have a nice antiviral effect. And even it was tested in phase 1A study, and one oral dose of 200 milligram was shown to result in a serum level that exceeds the inhibitory concentration of the drug with no treatment-related side effects or lab abnormalities. And now let's switch quickly to the other arm that is a potential antiviral, which is not less important than the first one, the immune modulation. And I would like to open and to note that both the innate immune response and the adaptive immune response seem to be somehow defective in the case of HBV. The HBV is known to be a very weak inducer of the innate immune response. It's considered to be a stealth virus that somehow escapes the innate immune response. And on the other hand, on the adaptive immune system side, the T cells, the cytotoxic T cells that are supposed to get rid of the virus, are somehow get in a state of anergy or exhaustion, and we do not really know what are the reasons for this. So let me give you one or two examples of innate immune response or target in the innate immune response can serve as antiviral strategy. So the STING is one of the interferon-stimulated genes. As you all know, there are more than 300 interferon-stimulated genes. Some of them are antivirals. One of them is the STING, which is a DNA sensor. It senses foreign DNA. And recently, it was shown that when you activate STING by a STING agonist, and you test it on a HBV-expressing mouse model of hydrodynamic injectant, you end up with interferon beta secretion, and more importantly, with a nice reduction in HBV DNA, and also concomitant with the induction of other ISGs like viperin, which is a typical antiviral ISG. The other player that is very popular nowadays is the TLR7. TLR7 is a pattern recognition receptor, PRR, that is present on dendritic cells and B cells. And upon stimulation of TLR7, those dendritic cells end up with secretion of many cytokines and chemokines, as well as interferon, thereby evoking both innate and adaptive immune response, something that seems to be like the perfect antiviral storm. So such a TLR7 agonist has been already tested in chimpanzees and has been shown to have a very nice antiviral response. And more recently, in a Wuchak model for HBV infection, it was showed that when you treat those animals, infected animals, with GS9620, you end up with a very nice suppression of surface antigen and even surface antigen loss. And in parallel, some of the animals develop anti-surface antigen antibodies. I think that one of the striking findings in this work was that the fraction of the animals that developed HCC went down very dramatically following this treatment. However, data coming from phase 1b trial done on patients in this case is a little bit less optimistic or a little bit disappointing. Given an oral GS9620 to HBV patients, those are two phase 1b studies, showed that there was no substantial effect on DNA, HBV DNA or surface antigen, but maybe the good news is that a fraction of the patients show some induction of ISG15, one of the interferon-stimulated genes that is known to be a target, one of the ISGs generated upon TLR7 stimulation. So maybe better later studies giving the drug for longer period of time or higher dosages will give us some better results. And last but not least, the T-cell response. As I noted before, there is some state of T-cell exhaustion in the case of HBV, and there are a couple of strategies that are trying to evoke or to wake up those T-cells in order to give them some renewed antiviral effect. One should keep in mind, of course, that whenever you do it, you endanger the patient with a robust immune response that can end up with, of course, massive cell death, overt hepatitis, and so on. So safety issue is very important here. So one of the ways to do it is a DNA vaccine. A DNA vaccine is a therapeutic vaccine that was introduced years ago, and here is an example of phase 1A study done on e-antigen positive patients following lamivudine breakthrough back then in 2006. We forgot those times already. And given the vaccination to the people resulted in some sort of immune response expressed as the interferon gamma secretion from T cells that were incubated with different HBV proteins. However, a very recent phase 1/2 study done on HBV patients who were DNA negative after cessation of nucleotide analogs showed that 5 IM injections of HBV, surface-based HBV DNA did not prevent the reactivation of HBV DNA that occurred in almost 100% of the patients, exactly as happened in the control groups. But maybe the solution is not to have enough with one strategy of immune strategy, not only DNA. Maybe the DNA vaccination should go with another strategy to evoke the T cells. So one of those strategies is to block inhibitory molecules that are thought to be responsible for the T cell anergy or T cell exhaustion. One of these molecules is the so-called PD-1, which is a very popular target right now in the anti-cancer therapy right now. So it might be not a bad idea to use it as an antiviral as well. And I think that this is a very nice study published in PLOS Pathogen in 2014. And the authors actually took a Wuchak hepatitis B virus model, and they treated the animals with a triple therapy, entecavir treatment, plasmid immunization, as is shown here, and also they treated the animals with anti-PD-L1 antibodies. And as can be shown here, there was a nice induction of immune response, and more importantly, there was almost, abolishment of viral replication. The DNA was down sharply, and also surface antigen was lost in some of the animals. So this brings me to the last slide, and to summarize, what we think that will be the future of HBV therapy. So I can only guess, but I think that the RT inhibitors, the NUCs, will stay and will be the cornerstone for treatment. But with no doubt, we have to add something else. We have to add some immune modulators in order to control the cccDNA itself. Maybe it will be aided by gene expression inhibitors like RNAi in order to evoke a renewed immune response. The entry inhibitors, with no doubt, will have a central place in order to control the spreading of the virus and maybe to have some indirect effect on the cccDNA as well. And last but not least, we will probably have some cccDNA inhibitors, either small molecules, either some drugs that target host factors that will be aided by host factors inhibitors that we are now revealing more and more to be important in cccDNA formation and biogenesis. I would like to thank you for your attention, and I will be happy to take questions.